Great, okay. I realize that I might be keeping you from lunch, so let's get started. I'd like to thank the organizers for the opportunity to present our work to you today. My name is Erin Kimbrel. I am head of the newly integrated Cell and Gene Therapy Research Unit at Astellas. But today, I'd like to focus on our gene therapy pipeline. This is my disclosure. So despite industry challenges, Astellas sees value in pursuing gene therapies. Currently, we're focused on AAV-based therapies in rare monogenic diseases. This is due to the very strong disease biology linkage. However, we are interested in leveraging this experience, building commercial capabilities, and then expanding into more common disease areas, such as polygenic diseases. And we really feel as though in this manner, we're going to be able to maximize the potential of the AAV platform. In order to realize this vision, we've been improving our manufacturing capabilities. This includes developing what we're calling our next generation process. This has enabled us to improve both our productivity as well as our yield. When we do this, we've been testing over 100 different constructs and multiple different serotypes and have found the NGP platform to be quite robust and versatile. In terms of productivity, we're around 1E12 VG per ml. And then in terms of yield from a 1,000-liter bioreactor, we're upwards of 3.4 E17. So the graph on the right-hand side of the slide simply shows one of our examples for overall yield coming from one of the serotypes that we're using. It's also important to mention that we have a high percent full capsid. We're over 90% right now. And so a lot of this work, put together allows us to really reduce our cost of goods as well as ensure we have the scalability to produce AAV vectors for not only rare disease, but also more common diseases. A lot of the optimization work that we do takes place in our South San Francisco facility, and then that is then transferred over to Sanford, North Carolina for our GMP manufacturing. So with NGP in mind, we're developing a broad and growing pipeline of gene therapies. So those that are shown in the red boxes are our clinical stage programs. We have 2 programs for XLMTM, as well as a GAA replacement for Pompe disease. A few of our preclinical and discovery stage programs are shown in the next 3 lines. We have myotonic dystrophy gene therapy program, as well as an Angelman syndrome therapy, and then we also have a neuroprotective AAV program for glaucoma. Of course, we also have some exclusive options for partner programs with one in Rett syndrome from Taysha, as well as one for frontotemporal dementia with Aviato Bio. So partnerships are really an important part of what we do. We're really looking for collaborators who want to help accelerate groundbreaking science across multiple therapeutic areas. And so what we look for in a partner is really goodness of fit, and that includes not only a pipeline fit, but also in capability and culture. And our current key areas of focus include neuromuscular disease, neurologic disease, as well as ophthalmology. Shown on the right-hand side of the slide are some examples of our recent partnerships. So in addition to the option agreements I mentioned on the previous slide, we're also partnering for access to different transgene designs as well as engineered capsids for better tissue targeting, such as our partnership with Kate Therapeutics as well as Sangamo. So now I'd like to spend the remainder of my talk really telling you about our own evolution and progress in X-linked myotubular myopathy, or XLMTM. So XLMTM, as you may know, is a serious, life-threatening neuromuscular disease that results in extreme muscle weakness. This presents as problems with both breathing and feeding. So XLMTM is caused by mutations in the MTM1 gene, which encodes a lipid phosphatase called myotubularin. Myotubularin plays an essential role in muscle development and maintenance. And so in a normal healthy tissue, you see nice thick myofibers with densely packed myofilaments and peripherally located nuclei. However, in the absence of myotubularin, the myofibers are quite small and you have a reduced density of myofilaments, as well as nuclei that have then migrated towards the center of the cells. So what this means for patients is just really severe muscle weakness. And what happens is that they need to have ventilator support or feeding tubes. In fact, 90% of patients will require respiratory support from birth, and that may include lifelong ventilator dependence for up to 24 hours a day. So it's really just a very terrible disease. And sadly, approximately half of boys that have a severe form of XLMTM will die before 18 months of age. So we've recently reported on our gene therapy program to treat XLMTM with our AT132 program. It's an AAV8-based gene therapy. And the findings from our Aspiro clinical trial were published in The Lancet back in 2023. In this trial, we compared both low-dose and high-dose AT132 against untreated controls, and with a primary endpoint of the ventilator support, hours of ventilator support per day. And what we found at both the 24-week and 48-week time point is that there was a statistically significant reduction in the requirement for ventilator support as compared to untreated controls in both the low and high-dose cohorts. Now, in these patient populations, there are impairments in motor milestones. However, in the Aspiro trial, we found that patients who were treated with AT132 had greater improvements in their motor function versus the controls. This includes tasks such as sitting up unassisted for 30 seconds or more, being able to pull up to a stand, as well as walking independently. And you can see the prevalence of these milestones in the treated versus control groups. Importantly, we also saw evidence of improvements in validated scoring systems for respiratory, motor function, as well as quality of life, as mentioned in the blue bar at the bottom. However, in this trial, there were significant safety findings. As you can see on the right-hand side of the slide, there were serious adverse events in both the low and high-dose cohorts, as well as in the untreated control. Sadly, there were also deaths in all three cohorts. Those in the treatment cohorts were associated with hepatobiliary disease and intrahepatic cholestasis, and the treatment-related serious adverse events also pertain to liver function. So as we reflect on these findings, there's really three key take-home messages. The first of which is that gene replacement of myotubularin can lead to significant changes in patient outcomes. The second finding is that, as an intrinsic component of XLMTM, there is intrahepatic cholestasis, and so we need to be mindful of the vulnerability of this patient population. And then lastly, high systemic viral loads of AAV may be something that increases the risk for these hepatic SAEs. So with the commitment to help find enduring therapies for the XLMTM community, we have been working hard to come up with a new next-generation investigational gene therapy for XLMTM. So this product that we're moving forward is called ASP2957, and it is meant to deliver a functional copy of the human MTM1 gene using a novel muscle-targeted myo-AAV capsid, which was developed by and licensed from Kate Therapeutics. So preclinically, we used an MTM1 knockout mouse to look at the impact of 2957 for improving motor function. And as you can see on the left-hand side of the slide, we looked at a rotarod latency test. And we tested various doses of 2957 from 1E11 up through 1.3 E13. And what we found is that with doses starting at 2E12 and higher, we saw significant improvement in motor function. What struck us about this is that the doses that we found for significant improvement in motor function are actually quite low and much lower than those from previously reported AAV-based therapies. AAV8-based therapies. So we confirmed muscle targeting through the dose-dependent increase in VCN and skeletal muscle, as well as through the expression in a dose-dependent manner of myotubularin. We're quite happy to see a very potent expression of myotubularin at levels that reached 100% of wild type or greater. This was also accompanied by morphological improvements in the skeletal muscle. We see nice thick myofibers and more of the nuclei moving peripherally as they should. So with these findings, we're moving back into the clinic to help our XLMTM patient communities with 2 different trials. The first of which is called Excel. This is a non-interventional natural history study where we're really looking to better understand and characterize the liver health and liver function within patients with XLMTM. The second trial is called Valor, and that is our ASP 2957 interventional study, where, given the preclinical findings that I showed you on the last slide, we're really hoping to be able to see some safety and efficacy from these lower doses than other IV-administered gene therapies. So this has cleared IND, and we're hoping for first subject dose later this year. Of course, first looking at safety and tolerability and hopefully some preliminary signs of efficacy as well. And so in closing, this has been quite a journey, and it's very important that we reflect on lessons learned and apply novel technologies, working with innovators, regulators, and patient communities alike. And so by really leveraging the findings from the Aspiro study and then moving forward, we're hoping to bring enduring value to patients with high unmet need. Thank you very much.